Hi, this is Dr. Jabali. And in this video, we're going to be talking about the topic 2.1, which is about molecules to metabolism. Here are some of the understandings, applications, and skills that we need to focus in this video. One of the first things we do, we're going to talk about what is molecular biology. We're going to talk about how carbon is so important for us in our elements, in our basically molecules inside of our body. We're going to talk about the 4 macromolecules that you definitely know them because you eat them on us every day, like carbs, lipids, which are fats, proteins, and nucleic acids. We're also going to talk about the metabolism, which is all the chemical reactions that are happening inside of us. We're going to talk about 2 ways, which is the anabolism, which is the building of stuff, and we're going to talk about catabolism, which is the breaking of stuff. Moreover, we're going to talk how urea was actually able to be made inside a lab and not from a living organism, which take away some kind of beliefs in the old times. And in the end, we're eventually going to be able to draw some of things such as glucose, ribose, fatty acids, and amino acids, and be able to identify them if those pictures come up in the test, you'll be able to spot them and know which one is which. So with that in mind, let's start with the first one. He says, basically, what is the definition of a molecular biology? That's basically we're studying everything. We're studying from their structures to their functions of this chemical substance, and we understand the interactions that they have with each other. Like right here, we have a picture of a sugar, right? This is what you actually see on a real life, but this is what we see in chemistry. All these bonds between oxygen, hydrogen, and carbon. And then again, we know that we need sugar, for instance, to be placed inside our mitochondria in order to produce energy. So there's always a link between all of these. And of course, each one has a different level of size. This is what we see, and this is tiny and tiny. But again, molecular biology, it's focused right here in the middle on all the bonds and interactions between these elements. As you can see, this is what happens. If you look at this picture, you will see that we have elements or basically molecules or compounds in everything. In the cup, you can see the coffee and the shape it has. You can see there's salt in the chips. And again, we have other things like vitamin C happening on the orange. But again, all of them have carbons, hydrogens, all these stuff. We are surrounded by these elements, and these elements somehow are connecting with each other. So first, let's recap a little bit of chemistry background. I know most of you guys should know the chemistry, but why not start from the scratch, right? So we know that we are made of atoms. And of course, there's atoms for each element in the periodic table, right? Hydrogen has its own atoms. The helium right here has its own atoms. Or for example, the boron here has its own atoms, and so forth. Each one of these elements has their own atoms. Now, what exactly does an atom consist? It consists of 3 things, right? We have the neutrons, the protons, and these guys are usually right here in the middle. And while we have the electron, which is on the outside. Another way of drawing this is usually they will have this as the nucleus right here, and then they will have shells coming out, and you have the electrons on the outside. Now, we also know that the protons are positive and the neutrons have no charge while the electrons have negative. But it's very clear that people need to understand that these atoms are very different depending on which element. So just to give you a little scoop here, a quick one so you can understand what I'm talking about. Just taking hydrogen. Hydrogen has 1 electron. So hydrogen will have its nucleus like this, and then it will have its electron. And that's it. That's a drawing of an atom for hydrogen. But for example, if you have oxygen in the case which has 16 electrons, it will have the nucleus, and then it will have 2 electrons here. That's 2, and then every other shell needs to be 8. So it's 1, 2, 3, 4, 5, 6, 7, 8. He has 10, and that means he needs 1 more, which is 1, 2, 3, 4, 5, 6. So you can see this is a hydrogen atom, and this is an oxygen atom. That shows how different they are because of the composition of their electrons. But that's chemistry. I'm just getting the basics of this because we need to know a little bit about these electrons. Okay, so here are the most 4 common chemical elements that we find in life, right? Carbon, as you can see, is a C, hydrogen, oxygen, and nitrogen. Chon, that's it, Chon. That's what we are about. We are basically, we have other elements in our body, but these 4 are the most common, and these are the ones that are going to build the macromolecules that we will talk later on in the topic 2, which of course is the carbs, it is the lipids, which is the fat, it is the protein, and of course the nucleic acids, which we're talking about, DNA and RNA. So these four are actually made a combination of these guys. Of course, there's more elements there, such as sulfur or phosphate. But the chan are the most common elements, and they interact with each other. So that means sometimes you're going to have carbon combining to an oxygen or carbon combining to a hydrogen. But once they combine in a certain amount of elements together, they form one of these four, which are the macromolecules, and this is what's the whole topic about. So before we get to the big molecules, let's talk about how they build up. So the first one we talk about is atoms of an element. When we say the same kind of atoms makes up an element, in this case, hydrogen, that's we just call element. It's just the same atom. Now, when you have a bunch of elements bound together, then it becomes a molecule. But again, molecule, if you notice here, it's still two elements. So for example, it can be oxygen with oxygen, carbon with carbon only, and so forth. It's just two elements bound together. Now, when it's different elements, now they're different, and they're coming together, we call it compounds. Again, this is chemistry. It's not necessarily for IB, but it's good for bio, of course. But you need to know. So element is just one. It's a hydrogen. Molecule, it's combining with the same elements, such as two hydrogens. And if I say compound, that means I'm mixing carbons with hydrogens, with oxygens and nitrogens, so forth. Okay? So basically, when we get to the point where we're talking about those big macromolecules, they're basically compounds. Now, of all these guys, we have to understand clearly how carbon reacts, because carbon is the base of us. It really is the base, and he's the one who holds up everything. And there's one little thing important about carbon that allows us to be together with our proteins, fat, is that carbons, they can share their electrons, they can connect with almost everything. So when we say share, we're talking about covalent bonds. Now, this is chemistry again, but let me explain why carbon is such a good thing, because carbon has this thing of being able to connect with other elements easily and then be able to share. And what it means by having four valence electrons. So here carbon, for instance, right? It has its nucleus with all the protons on and neutrons, and then it has its 6 electrons on the outside, as you can see here. Now we always know that the first shell here is always filled up with just 2, and then the next ones are 8. But out of the 6, 2 already went inside, so that means it's left with 4. So that means he has 4 valence electrons on the outside, and these are the ones who are going to connect with other elements. And most important, it will connect by sharing them. So covalent bond, for instance, we have carbon here. So carbon, usually we do the Lewis dot structure, which is basically we put the valence electrons on the outside here. Now, carbon only has 4 valence electrons on the outside. That means he wants to reach 8 electrons on the outside. That's the octet rule. He just wants to be sharing with everybody. So for him to get to a point where he has 8 on this valence electron, and again, if you don't understand what I mean, this is the nucleus, right? We have the first shell, which is always 2. They can fill up here. And the second shell always has to be 8, and then 8 again. So every element wants to get to that 8 because it means it just gets to be noble. That means unstable. So that means they just want to be in a cool, peace way. So we have here carbon with 4. So if I had to draw carbon, it would have been this. Carbon would have been, of course, we have the nucleus. And remember, carbon is made of 6, right? Electrons. So we have 1 and 2 on the first shell. And then on the outside, you have 1, 2, 3, 4. So that means carbon has 4 valence electrons. When I say valence electron, I am talking about the electrons that are found on the last shell outside. So he has 4 of those. So those 4 are not filling up. So what's missing here? We're missing 4 more in order to complete the 8. And those are the 4 more that he's going to connect with other elements. So for example, we have hydrogen, which is only made of 1 electron, right? This hydrogen. So for him to be complete, he needs that second one. This is where the carbon and the hydrogen says like, hey guys, let's just share, because this way you fill my shell and I fill your shell. So a hydrogen can come up here and share its electron. So what did they just connected here? They connected basically a covalent bond. They're sharing. It's not, and again, very clear, this is sharing. It means the hydrogen is not giving its electron away, and the carbon is not giving his away. They're basically saying, let's share ours. So that's right there is known as a covalent bond. They're just sharing each other. No one is keeping it. We're sharing both. But if you do this, now if you count the carbon, it has 1, 2, 3, 4, and 5. 5 is not enough. He needs 3 more in order to be complete. So what he can do is combine again with another hydrogen, and of course another hydrogen, as you can see here, right? This is connections. So right now you just created one compound in which the carbon has fully now the H shells because it's sharing with these guys. If I had to do a nice little drawing of this, what it would have looked in a structure, it would have been attached like this, and then like this, and like this. So I just created a perfect compound right here that is stable, right? Because the carbon, it has basically filled up its last shell, so that the hydrogens, and they're sharing their electrons. That's a connection. Now, this is exactly how we go from something so small like this to be combining bigger ones. And that's where we can do the protein, the lipids, the carbs, and so forth. So you can see here more examples of connecting the carbon with others. So we just drew this one right here, basically, which is this. And you can see you can add more carbons connecting with hydrogens. You can have other ones with double bonds or triple bonds. These are just covalent bonds. They're sharing, electrons, and they're building molecules. And by going that way, we can reach to what we call the organic compounds. And when I say organic here, it basically means that it actually has carbon. So right here, you can see now there's a lot of more joining of hydrogens and carbons, sometimes oxygens, and so forth. These right here are what we call the macromolecules. And what I mean by macromolecules, I'm talking about the molecules that we have in our body that we need on every single day. They are the carbohydrates, the nucleic acids, the proteins, and the lipid, which is basically the fat. And you can see their structures right here. You can clearly see the carbohydrates are usually rings. And then amino acid, you can say that it has an amino group and a carboxyl group. And these are the ones that you actually have to know how to draw. The nucleic acid, the phosphate, the oxyribose, as well as the nitrogen base. And here you have 3 fatty acids with glycerol. Now I'm just talking quickly, but we have in the next videos, it will go more in depth about each one of these guys. But for now, I just wanted to show you how this all started just by carbon wanting to be in covalent bond with other elements and basically sharing. So it's all based on the carbon. So moving on, let's look exactly in what kind of carbohydrates we're talking about. Well, carbohydrates are basically known as the sugar, right? And we're going to be drawing more specifically in detail the sugar stuff. But basically, it will look more like a hexagon, okay? This is what the sugar of a carbohydrates looks like. And of course, as you can see, it has more details if you see right here with the oxygens, the hydrogen stuff, which you will eventually have to draw, but I'm not going to do it at the moment. But the interesting about carbohydrates is that it has one as a monomer. We call it monosaccharides. And the word saccharide here means sugar. So that means it has one sugar. But the interesting about carbs is that carbs can actually connect with 2, with 3, and so forth. So in the case here, carbohydrates, they can have examples of monosaccharides, for example. Let's put monosaccharides right here. We can have glucose, we can get fructose, and galactose. These are all just one unit of sugar. They can build up to make 2. If it's 2, then we call it disaccharide. So these are 2 units. In this case, we can talk about sucrose, which is the table sugar. We can talk about lactose, which you find in the milk. And we can talk about maltose, which you can find in spaghetti. But these are made of 2. And then you can do polysaccharides. Let's see here, caride. And in this case, sorry for the handwriting, being like a doctor, it actually happens like this. Disaccharide will be like a lot. That means we have basically 3 or more sugar units. In this cases, we will have things like starch, glycogen, we got chitin, I call it chitin, but you can call it chitin, that's the right way, and so forth, and so many other ones. So these are just examples of carbohydrates, which we will get more in depth when we go to the videos on carbs. But these are just building blocks of carbons with hydrogens and oxygens, and it can go from one to become many, okay? Then we have the protein. Protein also has carb on it, but it not just has carb, it has hydrogens and nitrogens and oxygens. Now, the most common drawing of a protein in this case is that you start with a carb in the middle, and then the carb will be connected to other things. So in one side, the carb can be connected to a nitrogen with a hydrogen on the sides. And then on the other side, it can be connected to another carbon. It can also be connected to a hydrogen here and down here to the R variant. This guy means I can be substituted. That means I can interchange with different types of structures. So usually we call it the variant. Okay, and then this carb here will be connected to an oxygen and connected to a hydrogen. Now, we have to know this by heart. You have to know the amino acid. This is amino acid, by the way. Amino acid is one piece of a protein. What does a protein looks like? Protein is like a bunch of amino acids together. So it's like, let's put it here. If this is amino acid attached to another amino acid, attached to another amino acid, attached to another amino acids. So each one of these guys are an amino acid and they're connected by a bond. But first, let's look back into this amino acid. We have here this structure, which is known as carboxyl. This is in a carboxyl group that is always on the right side of the amino acid. And we have it here on the left side, the amino group. So this is a skeleton, a structure that is firm. And what I mean by firm, as in it will have all this section here firm all the time in every single amino acid there is. The only thing that is changing in an amino acid is the R. And that's why we have 20 different amino acids. But what changes is always the R. The rest is exactly the same. So if you memorize the green part, you're good. Now, just to be clear, this is one amino acid. But if I had to make it zoom out, I would say this circle here is an amino acid. And it's attached to another amino acid. The bond here is known as peptide, right? And then together, because we have two here, we can say dipeptide. We call it a dipeptide, not because there's two peptide bonds, but we call it dipeptide because di means there's one amino acid, there's two amino acids bound by a peptide bond. And when I say peptide bond, that is a bond only found in proteins. And then we can have a third amino acid that will be tri, right? We will call it tripeptide. And then many more other amino acids. And we just keep bonding them with peptide bonds. Eventually, they can become polypeptide, in which the word here means many. So we have many peptides because we're bonding a bunch of amino acids. That's protein. Then we have lipids, which again, lipids, in another word, a common word is fat. Now, when it comes to lipids, there are so many different forms of lipids. Examples. So let's go through some examples. We have phospholipid, which you probably already learned on topic 1 when you were doing the cell membrane. We can have cholesterol. We can have lipoprotein. See, these are just small examples. And there's one that is basically the fat, or what I would say the bad fat, which is known as triglyceride. Triglyceride. Now, the triglyceride right here, this is the one structure that you really have to know besides, of course, this one. These ones you can just recognize. You should be able to draw a phospholipid by now if you did topic 1. Triglyceride is basically made of 3 fatty acids plus 1 glycerol. So basically, it's like you're putting a structure like this. 1, 2, and 3 fatty acids connected by a glycerol. So in this case, you can see glycerol is this section, while the 3 fatty acids are these guys. This is what a triglyceride would mean. It's the fat that moves around. Of course, we're going to have a video just on lipid and get more detail on its structures. But these are examples of lipids. Then we have the nucleic acids. That is the famous DNA as well as the RNA. Okay? And again, these guys also have carbons, hydrogens, nitrogens, even phosphates. So they usually have a structure with a phosphate group attached to a sugar and then eventually attached to a nitrogen base. As you can see, that nitrogen base, that means it has a nitrogen. Here we can have the oxygen, there's carbons and hydrogens here, and here's phosphates with some oxygens and hydrogens. So you can see it's a lot of carbs and hydrogen, but all of them, if you notice, all of them, I'm showing that there is carbon in there. Of course, I will go more details about the structures of these two when we get to its video. But here is just giving you an example of the 4th macromolecule that needs carbon in order to go through. So here is what eventually the entire topic 2 is about. It's just starting from the basis, which we're starting with its structures, right? For each one of them, amino acid, nucleotides, monosaccharides, fatty acids, and then you build them a little bit bigger. So for example, for the sugar, as you can see here, let me erase this so you get clear. You can see that it goes into becoming a sugar, small sugar becomes a big sugar, which is a starch. And then that starch basically can be produced in the chloroplast. So you're going from the smallest molecule to the big thing. Nucleotides, for example, they make up DNA, and the DNA will make a chromosome. Amino acid will become a polypeptide, and this guy will become filaments, and it can actually construct the cytoplasm or muscles and so forth. Fatty acids will try to triglycerides, and these will become adipose cells, which the word adipose means fat cells. So you can see we're starting from the smallest thing and in direction towards the big stuff. So that's why it's very important that you learn the smallest things so you can understand how we're building the big ones. So this goes back again to wrapping what is organic compounds. And it's any compounds that contain carbon. And when I said compounds, remember, compounds are not the same elements. It's a mixture of different elements. So most, and I will say all, but most of the compounds which have carbon mixed with hydrogens are considered as organic. Of course, there's some exceptions. And usually here, these guys are the exceptions, like carbon monoxide, carbon dioxide, carbides, carbonates, cyanides, cyanides. These guys are the exceptions. They do have carbon, but they're not organic. But the ones that we're going to focus as the organics is the protein, the carbs, the lipids, nucleic acid. These are 4 examples of macromolecules that are organic. So when I say organic molecule, or if you read in the IB, any question says organic molecule, we are assuming that it's protein, carbs, lipids, or nucleic acid. Keep that in mind. Now, there's a part about vitalism which you have to know. Vitalism is like saying that it's a doctrine, okay? It's basically, they believe that physics and chemistry alone, these two, could not explain some of the process of life, that there was some magic out there. There's some kind of non-physical inner force or energy, more like chi or prana, or whatever you guys want to call from the East Asia, which they have a lot of these beliefs, which it's actually coming back now. There's a lot of beliefs coming out. This is basically saying that not everything can be explained through physics or chemistry. There's some force out there that we cannot see that actually takes charge in building up these organic molecules. But vitalism believes that organic molecules could only be synthesized by living system. Okay? They thought that it had nothing to do with inorganic molecules. That means it cannot just come from a carbon and then boom, you become an organic molecule. But there was one experiment, which you have to know because IB might ask you this as a multiple choice, is there's one experiment that disproved vitalism. Because again, what does vitalism says? Hey, there's no way that you can make an organic molecule from inorganic. Well, this guy, Frederick Wöhler, he did. And he did by mistake, by the way. He was just in the lab working his way, and out of the blue, when he was actually heating inorganic, okay, salt, in this case ammonium cyanate, it turned into urea. Now, for those who don't understand what urea is a toxic that comes out while you pee. So every time you pee, urea is coming as a waste product. And if you keep holding your pee, you're holding urea. Urea is very toxic. That's why we have to pee every day, because we gotta get rid of this urea thing. So they think, oh, humans, other animals pee, that means we produce urea. So we think that urea can only be produced by something like organic, because we are organic, right? We have carbs, we have lipids, we have protein, which is all organic. So they thought, oh, urea can only be produced by something that is organic. So it can only be produced by us or other animals. Well, this guy, Frederick Willard, said, no, my friend, that's not true. Guess what? In the lab, by mistake, I created pee. And I did not have a human creating, I had an inorganic material creating. So that clearly, what they did was just simply erase vitalism. The idea that everything comes from something else, or more important, organic material can only come from organic. It just proved that it can't come from inorganic. So now there's this thing called mechanistic materialism, which believes the opposite. It actually says that every life there's in the world actually came first from an inorganic matter. And that eventually, with time, starting from elements such as carbs, hydrogens, all these guys, they built up and they made up the big ones like us, or the mitochondria, or the cells. So all started with the elements. That's what mechanistic materialism view is at the moment. So, do you have to know the whole process? No, you don't have to know the whole process of how the lab was done. So you don't need to know all of these guys and how it's produced in the body, okay? Because this is basically what's produced in us. But you do need to know that it was cyanic acid and ammonia, which is ammonium cyanate. That's the inorganic salt that was in the lab produced urea. So they might ask this part. So if you have to memorize, it's this one that you need to know. The one that it was produced in the lab, coming from this inorganic molecule to make this organic molecule, okay? Now let's get to metabolism. Now, metabolism is basically all the chemical reactions. You can see there's like a lot of reactions happening in our body, right? And all these chemical reactions that is happening, they're building us, they're helping, giving us energy. So all types of breaking downs and making ups inside your body is known as metabolism. So we have many of that. And for those stuff, we also need enzymes, which we will learn this more on topic 2.5. But basically, it's just all the chemical stuff that is going inside of our body, and all of them need enzymes to break it down. So for example, for you guys to have a scope a little bit, because this is not topic 2.5. So for example, lactose. Lactose, you find it in milk, right? It's a type of sugar that you find in the milk. You need an enzyme, which is known as lactase. The lactase breaks lactose into glucose and galactose. So it breaks down into two pieces. If you remember, lactose is made of two sugars and eventually splits into one sugars. So lactase is just one enzyme, but if you think about it, we have about 1,300 enzymes in our body. So you can imagine so many different chemical reactions that are happening in our body. So what is the main point of metabolism? Why do we have metabolism? There's two reasons why we have metabolism, and you need to know. One is to give us energy. So for every time, for example, when we're eating food, that's a chemical reaction, right? And what you're doing is you're breaking down the carbs, the lipids, the proteins, the nucleic acids that you find on food. And why do we break them down? To get energy. So basically, if you want to grow, if you want to produce, if you want to talk, whatever it is, you need to make energy, right? That's one. Another one is to build things. So you break sugar, but why did you break sugar? To make energy, okay? But sometimes you get proteins. Like you eat, a lot of people say you want to go to the gym and you want to pump it up and you want to build muscles. Well, building muscles, it's part of the assimilation. Why? Because you're putting the proteins together, and those proteins together makes your muscle. So synthesize here, the word synthesis, it means make. So you not only are breaking down food to get energy, but also you want to grow, you want to make more muscles, you want to build up your bones, you want to get extra, God knows, hair coming out of you. All of this stuff needs things to come in and assimilate, okay? So, or you want to make more enzymes, you have to build proteins, whatever it is. in your body, you are not only breaking things, but you also are making things. So that's why we have metabolism. It is for two, to make and to break. And with that, we talk about these two. The catabolism, which is actually to break things down, and anabolism, which is to make things up. So for example, catabolism here, you can see this is a large molecule. And what happened is, it is breaking down into small little pieces. So it's just like you eating food, right? You're breaking the food into small pieces and you're picking up the energy from it. When it comes to anabolism, you come in with small molecules and you're going to join them to become a large molecule. And this can be an example as building your muscles for simulation. So when you combine in your body all the catabolism and all the anabolism, that's metabolism. So you have to know, IB asks you to know and compare as well as contrast the differences between anabolism and catabolism. So first, let's look at its purpose, and then we're going to look at its energy and an example. So for purpose, of course, anabolism is to make big molecules from small ones, while catabolism is to break the big molecules into simple ones. The next one is energy. Well, if you want to make something, right? Even let's say if you want to build a house, what do you have to do? You have to use your energy to put those bricks up, right? So you need to use energy to make something. But if you want to break something, if you're going to release the energy that was held up inside of the whatever molecule or whatever product you were using. So again, anabolism uses energy to make, while catabolism basically breaks the thing and releases energy out. Now, example, photosynthesis. Photosynthesis is an example of anabolism because photosynthesis is making sugar, so you're building a sugar. Cell respiration, which is basically us eating our food, that's the opposite. We are breaking the food to get the energy out of it, so the energy is being released out of the food. And you can see the pictures right here. This is an example right here of anabolism. Actually, sorry, that's an example of catabolism. Why? Look, here we have completely joined molecules, right? They're all joined by the bonds and then break into pieces by themselves. This is catabolism. Now, why I see water here? Because every time you want to break something, you have to add water. That's why we tell you, if you want to lose weight, my friend, drink water, because water helps break down the food. The more water you drink, the more you break down the food in your body. Here is an example of anabolism. The pieces are alone and then they combine together. But if you look clearly, let me take this part, look what's happening. Water is being released out. So that means every time you build things, water is coming out. And it makes sense. Like when you're working out or building on something, water will evaporate. So clearly, we have to understand that this is part of IB, and this can be easily a question there where they ask you to compare anabolism and catabolism. So as an IB examiner, I would say do know the difference between them and also know an example for each one of them. As I have mentioned here, example photosynthesis for anabolism and cell respiration for catabolism. Do know these examples because they are perfect and IB will always accept them. And have at least two differences between them. One talking about they're breaking down or making, and the other one is where's the energy going, okay? So let's go more in details for each one of them. First, anabolism. Another word for anabolism is known as condensation reaction. Not only is condensation reaction, dehydration synthesis. So these three words means exactly the same thing. So if you see this in the IB exam, know that the three of them are exactly the same thing. Anabolism, condensation reaction, dehydration synthesis. These three words are talking about the same thing. What are they talking about? Building something. They are all about build. Now, for them to build this, they will have to get two molecules together, right? You have a molecule here and you have a molecule together, and you're going to build by connecting them. Now, to connect them, something has to come out of it, and that will be water. We're going to look exactly at an example because you have to be able to draw an example showing that. So every time you build something, water comes out. That's why if you break this word, dehydration. So the hydration here means water, and D means off. So off water. So whenever you want to build something, water is coming out of it. That's a way to remember, okay? So once again, dehydration synthesis, condensation reaction, anabolism, the three means building something. And as you build, you are releasing water out. So here is an example that you might be asked in IB, which is to show an example, and this example has to be done with structures such as the amino acid. So that means you should be able to draw the amino acid by scratch. They will not give you this picture that I'm showing right here. You have to know it by heart. So right here we have two pictures of amino acid. This is one amino acid that is going to bind to another amino acid. Now, if you see what's highlighted in red here, is the things that will come out, right? So you can see there is a hydrogen oxygen come from here, and there's a hydrogen from another one, and these guys are going to come together. So when they're going to become what? These hydrogens are going to become this water right here. Like I said, water is coming out when you're building something. And then there will be a connection that will happen between these two molecules, which is right here. It's the connection that we have here. So I'm going to draw this from scratch, and you should be drawing with me because you have to know it by heart, but this is just a view for you to see. So right here, we are doing what we consider as the dehydration synthesis. Okay, so dehydration synthesis, once again, is about building. So I'm going to put it here just as a reminding for you. It builds. So you do have to remember amino acids. I'm going to take an amino acid and bind to another amino acid. But you cannot put this. In the test, if you put just circles, you're not going to get point. You have to draw them in details. So me drawing an amino acid here, we have a hydrogen connected to a nitrogen connected to a hydrogen. Right here, what I'm doing right now is building one amino acid only. That's what I just did here. I built that one amino acid. Then I'm going to put a plus here because I'm saying I'm about to add with another one. Then I have to draw again another amino acid. With the hydrogen, the R, carbon here, bound to oxygen, OH. Forgot the OH here. All right, now we have finally built the two amino acids. Now you need to show the next thing. The next thing is the arrow. You have to put an arrow because you're showing that this is a chemical reaction that is happening. Of course, you don't have to have another color in the test, but if you do, then what you do is you would circle up the OH with the hydrogen of the other bound like this. Because this is showing that where you're getting that water from these molecules, right? And these molecules, you can always go and say that is the water that is going to come out like this. That plus what? What's coming out here is, let's continue in black. So we have a hydrogen with a nitrogen with a hydrogen. Again, this is tough, but the only way you're going to memorize this is if you draw it many times. Repetition works, people. And then you're going to have the carbon here. And here's the interesting part, is that this carbon right here is the one that is going to connect. Again, let me highlight here for you to see. This guy is going to connect with this guy. So that means the bond is going to be right here. It's going to be between this carbon and that nitrogen. Okay? So you connected that bond, and then you continue with the nitrogen here. You can have the hydrogen here, and carbon with the hydrogen. Oops. Carbon with OH here, and hydrogen here, and the R. So then now you have built an amino acid with another amino acid. That means you have built a dipeptide and the water is out. Now, to get the extra point, not only did you mention the title, you build the two amino acids, you put the plus, the arrow, and you have the water coming out. You can also show here the label of the dipeptide bond, which is this. What is this bond here? That's a peptide. That's a peptide bond right here. Because remember, the bond that we call for proteins, or should I say between an amino acid and amino acid, it's a peptide bond. So you just have to connect it. Now, this is an example of dehydration synthesis that is built. You draw this and you get a full point, because your point first is that you have, let's put another color here. Let's put in red here. First, you have drew an amino acid that is perfectly right. Second, you have shown the water has come out. From where did it come out? You have shown an arrow. You have built the entire amino acid together and you have labeled the peptide bond. You have to do that. You probably already got yourself about 5 points there just by doing all of this. Okay? Now, what we did was building. Now we're doing is breaking things down. When we break things down, we call it catabolism, as well as hydrolysis reaction. So the word hydro right here, it means water. And lysis means cut. So we're cutting something with water. In this case, that means water is cutting a product. Or should I say a molecule, or whatever it is, a thing. So remember, hydro cut, so that means water is cutting something, which is catabolism, because catalysis means the breaking down of something. And you can only split something or break down something is by what? By adding of water. So when you add water, the water is there to break things down. Why is that? We'll see here in the process where you're going to see that the oxygen, the hydrogens of the water will be splitting into what we call the hydroxyl group and the hydrogen ion. So one is going to take a hydrogen and the other one's going to take OH. And you should be able to draw this too, because they might ask you, instead of drawing the dehydration synthesis, they might ask you to draw the hydrolysis in this case. So let's do the hydrolysis, right? Okay, so hydrolysis in this case, as we were saying, this one you're breaking things down, right? So we are just going to put here break down, so you can remember it. Now, breaking down means they were already a big piece. So we have to draw the entire, for example, I'm using amino acids, of course, in this case it's dipeptide. We have a nitrogen here, carbon. Again, be very clear on your drawings. Oops, this one is not supposed to be there. See, I already started making a mistake. Oops, there you go. There's a carbon with a hydrogen bound to a nitrogen with its hydrogen bound to a carbon with its stuff, OH and H. See, this is a piece of dipeptide. That means it's built up. The point is, you want to break it. So if you want to break it, you add what? You added water. So you have to show water plus the dipeptide molecule, right? When you do this, then you have the arrow here, which is going to show what? You're going to have the hydrogen molecule, carbon, hydrogen, the R, and then the carbon H. You're breaking the piece, and you have the nitrogen here, H with the R, with the CHOH here, plus the H. I separated, right? Where does the separation happen? Right here. This is where it was broken. So you can even put an arrow if you want. What's happening with this arrow is the water is the one that is going to this arrow. So the water is basically made of this, right? So the OH here is coming down here, and that H is going for the hydrogen. And voila, you have the two amino acids that have been separated because you added the water. So this is about add water. You can write that in the test, you get points by saying, oh, it's by adding water. And what are you doing? You're breaking down the peptide bond that is right here. So this is an example of a drawing for hydrolysis. Once again, you have to know both ways. You're not going to get points if you don't do this. Now, is drawing it enough? No, you can put some annotations there. Like I said, breaking down by adding water, this can be points there. You can put arrows also showing the way in the pathways. Okay? So with that, we have examples of hydrolysis. Like I said, hydrolysis, and I'm going to repeat this, it's breaking things down, and it's about adding water. Now, look how interesting this is. If you want to break down glucose in this case, right? I mean, the lactose, which is milk, right? The sugar in the milk. If you add water, it would turn to glucose and galactose. Now, for this, how many water did? It was just one. Now, if you want to break down the starch, what do? You put just one water and you get a bunch of glucose. Now look at fat. For one fat, you need 3 water. This is what I'm telling you. If you want to lose weight, if you want to break down that fat, drink a lot of water because you need a lot of water. And as you can see here clearly on this picture that I'm showing here, that you need 3. For this, you needed 1. This is none. So for every single, even for protein here, look, it's something that you need a lot more because water is that's why it's very hard to break down all our muscles. It's the last thing that happens. But when you have a lot of fat, The more you drink water, the more likely you're breaking down that fat. Now, we usually break down sugar first. That's why water is so fast, that's why sugar is so fast to be broken down. Fat is the next one to be broken. That's why amino acid is the last one. So again, carbs are the first one to be broken in your body because that just needs one water. Fat is the second one, but it needs three waters. And then proteins are the last thing to be broken in your body because it needs a lot of water. So why not drink a lot of water? Just in this case to get the fat, because to get to the point where we're breaking proteins, that's gonna take more than 5 gallons, man, 5 liters, and only if you're doing a lot of sports, man. But anyways, so here is another example of structured drown. It can be done also with sugar. It can be also done with fat. This is an example of a sugar structure showing and becoming 2 pieces. And here's the fat with the water breaking into 2 pieces. You don't need to know these guys and how to draw them. Just stick to the amino acids, then you'll be fine. And this is pretty much it. Here is all the questions that you can find also on the study guide sheet for you to go over. You can go back and watch these videos and you'll be able to answer these guys. More of these guys again. And that's pretty much it. Thank you.